Great. Well, thanks, Kate, for inviting me, and it's really an honor and a privilege to be speaking to you all here virtually. I wish I were there in person in a lecture hall at Montana State. I probably ought to say at the outset that I am a proud MSU Bobcat parent, and I read virtually every missive that Dr. Cruzado sends out to parents. So I'm pretty in touch with the MSU culture just by virtue of what I read from the leadership there at Montana State. And so that's helped me frame this poll that I wanted to do um, as to what's motivating folks out there to attend this talk. Now, I can't do a formal poll, so I'll read out the choices. I hope most of you are here because you're interested in the subject, but I totally understand based on what I've read from the teachers and instructors at Montana State that some of you may be here for credit for your honors college class. I personally, if I were an undergrad, I'd be there for the champ change, although Kate says that's very overrated. So I guess that's probably not really cool to say that I would want the champ change, but I think I would anyway. And then lastly, there's going to be some on this call who are related to me. And so I hope that their questions are more professional than personal over the course of this talk. But um, hopefully, if we did a poll, that we'd see a nice bar for A. Now, before we dive into the concepts of precision medicine here, I'd like to do a little bit of a background introduction of the topic and how I came to become very passionate about this field. I was fortunate actually to get started in the field of genetics back in 1991. And at that point, one of the landmark developments in genetic technology had just been developed. It was referred to as polymerase chain reaction, and it gave us the ability to probe into the field of genetics like never before. And that fascinated me, that ability to really peer into the molecular world. Fast forward to my time at Stanford as a fellow in reproductive endocrinology and infertility, which are the physician specialists who perform in vitro fertilization. And at that stop in my education, I became acquainted with the whole field of genomics, which is genetics on a larger scale, and the application of genetics to help tailor what we do in medicine. So with that as a background, let's dive into the topic of precision medicine. If you have a question, please feel free to interrupt me, and we will handle it as we go. I'm sensitive to the fact that this is later in the day there at Montana State, and you may have other things to do at the end of the talk. So I want to make sure we address your questions real time as we go through the topic. So with that, let's dive in.
This is going to be completely unprecedented. It is going to affect medicine in ways we've never seen before.
You can match a blood transfusion to a blood type. What if matching a cancer cure to our genetic code was just as easy, just as standard? And that's the promise of precision medicine, delivering the right treatments at the right time, every time, to the right person.
And that State of the Union address really launched the field of precision medicine in a formal way in our country. Prior to that, we had referred to this field as personalized medicine. And there are subtle differences between those two terms, but by and large, they are synonymous. And really, the definition, according to the National Institutes of Health of precision medicine, is an emerging approach for disease prevention and treatment that takes into account people's individual variations in genes, environment, and lifestyle. So many of us think of precision medicine as being something that leverages the specificity of our molecular signatures to develop individualized approaches to our individual healthcare. And the motto of this field is typically the right medication for the right person at the right time. And that specificity of our molecular signatures allows us to develop new approaches to disease in terms of classification, diagnosis, prognosis, as well as new treatments. The State of the Union address in 2015 was very significant. It was the year that former President Barack Obama rolled out the Affordable Care Act. But instead of using the time on healthcare to unveil the Affordable Care Act, former President Obama used that bandwidth in his State of the Union address to unveil the Precision Medicine Initiative. And the Precision Medicine Initiative is a significant sea change moment in the way that funding occurs for this particular field of medicine. A total of $215 million was earmarked for the launch of the Precision Medicine Initiative. And academic institutions across the country used this as a signal call for a change in their structure to embrace, support, and develop this emerging field of medicine. But is using our individual variability, the differences between those of us that are on this WebEx call, is that leveraging those differences, is that a new concept? Well, actually, it's not. Leveraging individual variation for healthcare benefit is a very old concept, actually. And it was really heralded by Landsteiner's discovery of the three basic human blood groups. And as most of you know, we all have our own individual blood group, but we share it among one another. Some of us are A positive, some of us are AB negative. But Landsteiner actually originated the ABO system. And then that ABO system that designated differences between patients was leveraged by Ottenberg to perform the first matched blood transfusion in 1907. Over 100 years ago, precision medicine was really already in practice. In this day and age, we don't just match blood for the basic groups of ABO, but we also match for Rh factor, as well as tens of other antigens that are present on blood cells to ensure that we don't have a transfusion reaction. So what is different? What is new? What makes precision medicine a new and heralded concept in medicine? Well, these are the signature developments that have really ushered this day and age as being the age of precision medicine. First, large-scale biorepositories or biobanks and biologic databases are being developed. And one of the most important in our lifetimes was the Human Genome Project, which was the first sequencing of the entire human genome, all 3 billion base pairs. The next important development towards really making this the era of precision medicine is the development of very high-powered molecular platforms to characterize our molecular differences. We're going to talk a little bit about the omics later in this talk. Finally, and just as important, is the development of computational tools and expertise, collectively referred to as bioinformatics, to analyze these huge sets of data that are in the terabyte range of volume. So those three developments are really what makes this, rather than 1901 to 1907, the age of precision medicine. And we're going to talk about how this has transformed our approach to the practice of medicine. But before we do that, we should go back to basics. I never presume how much genetics my audience has as a backdrop. And the reason I don't do that is even medical students and residents that I train at my hospital are not real familiar with genetics and genomics. So I give the back to basics even to those that are in the medical profession. So let's Let's actually go through that a bit, because with that background, you'll be more aware of some of the concepts that I'm discussing throughout this talk. I don't know why this cell is green, but it starts with a cell and a nucleus, and within that nucleus are all of the chromosomes. Chromosomes are the units of DNA that contain a variable number of genes, which are really the reading frames of our genetic code. Some of our chromosomes are packed with genes, like the first chromosome, which carries 2,968 genes. The chromosome with the least genetic baggage is the Y chromosome, only carrying 231, but carrying a very important gene known as the sex-determining region of the Y chromosome, or SRY, that delineates male from female on a genetic basis. Genes are those segments of DNA, those reading frames, that are transcribed to RNA, which is different from DNA, and translated from nucleic acid to protein. A base is a basic unit of DNA that has a pairing associated with it. So you've probably heard of the CTAG system of DNA. Cytosine is the C, it's always paired with guanine, and adenine is always paired with thymine. And that makes up the code. We have 3 billion of these bases and base pairs within each of the nuclei in our body. Just kind of crazy to think about in terms of the scale and volume of genetics. Now the concept of a single nucleotide polymorphism, and you'll see in this cartoon that there are several of those listed here. A SNP, or a single nucleotide polymorphism, is what makes us different in terms of our code. It's a variation at a single base pair that occurs at a specific position in our code. In order to be classified as a SNP, it has to occur in over 1% of the population as a variable. So not every change in a base gets the title of SNP, but these are regions of variability within our code that make us different from person to person. The genome is the entire set of DNA, all 3 billion base pairs that are contained within the nucleus. So let's talk about the central dogma of molecular biology, because we really got into DNA there and its relationships within cells. But this talk will not just discuss DNA, but also other layers of molecular medicine, including RNA and protein. And this was originally described by Sir Francis Crick in 1950. And I actually had the opportunity to meet Sir Francis Crick when I was in England, and that was a high water mark for me. But he is the originator of the central dogma, which is what basically describes the DNA replicating itself when new cells are developed mitotically. Going to RNA, that's actually going from a double strand to a single strand, and that process is known as transcription. And then finally, the single-stranded RNA gets translated to protein at ribosomes, and that's called translation. And so those are the 3 layers of molecules that we'll discuss through the talk. And these cartoons or icons that I've created for these different layers are going to be kind of one of our touchstones throughout the different platforms that we talk about. When you see one of them, you'll know what the platform is designed to detect, because some of the tests in medicine detect protein. In fact, most of them do. But some of the tests detect RNA, and some are actually designed to detect differences in DNA. But that's the central dogma. It's the foundation of molecular biology. The DNA is static. In other words, it really doesn't change from cell to cell. Sometimes you get mutations during the process of cellular division that may render some changes, but by and large, our DNA is fairly static. It's who we are. It doesn't change much. On the other hand, RNA and protein, they change a lot. They change as a consequence to different exposures on the parts of our cells. And they are a whole different layer of molecular complexity that may actually provide an opportunity to diagnose different types of exposures and different disease states. So we'll come back to that through the talk. How does the central dogma apply to precision medicine? Well, on the left here, we've got an unexposed cell, and you'll see that the green part of the DNA is actually going to be the part that gets transcribed into RNA, translated into protein, and then actually picked up through capillaries into the bloodstream to be detectable. In an exposed cell, you may get a different part of the DNA that actually gets transcribed to yield a very different RNA that yields a very different protein that when it is out in the bloodstream may be detectable and readable in a report to allow us the opportunity to diagnose a condition of exposure. So imagine that exposure may be a radiation to a cell that actually has generated a new protein product, or maybe it's a different nutritional exposure, or perhaps it's an aging cell. Whatever that exposure might be, the hope is that it will be something that is detectable through an assay that is non-invasive, such as a blood test.
5 years ago, it was about $10,000 to sequence a whole genome. 10 years before that, almost $1 billion was spent worldwide in sequencing this. Today, we can sequence a whole genome in 24 hours for $1,500.
And that really describes why we are on a really expeditious course in precision medicine, is the pace at which technology has made these developments scalable. It's hard for me to believe that the Human Genome Project, which cost $3 billion to achieve, now can be done on my benchtop platform in my lab in a matter of 24 hours at a cost of $600. And that's a 20-year journey to go from $3 billion to 600. That easily outpaces Moore's law, which was originally applied to electric transistors, talking about how quickly they develop into better products. You can see the pace of sequencing in terms of how it's led to these cost reductions for probing into our DNA and RNA levels. Many of us are probably familiar with some of the direct-to-consumer tests that are available out there. And over the holidays, I saw a lot of Ancestry.com advertisements, but the other company that you see a lot of adverts for is 23andMe. These are direct-to-consumer genetic testing companies, and the FDA has had quite a journey with them. They originally were available to the public in 2006, and now the FDA actually regulates them depending on the application. They have an important role to play in terms of how people approach their healthcare. And an article that appeared in 2017 looked at the impact of these direct-to-consumer tests on how patients approached their medications. And what's interesting about this study of 961 people who used the 23andMe system is that 91% of the people that used 23andMe got readout from the company that they had at least one result that was consistent with an atypical drug response. Now, that's kind of alarming to people that don't really understand the genomics and genetics that underpin these tests. And of those 91% who were told that they had an atypical drug response risk, 5.6% of patients actually changed their medications on the basis of what they got from the direct-to-consumer testing. Unfortunately, a handful of those actually changed their medications unsupervised, and that is can be problematic and very risky for patients to do. When you have 91% of consumers being told that they may have a genetic predisposition to an atypical drug response, that's going to generate a lot of questions. Is our healthcare system ready for all of those questions? Well, U.S. News and World Report was encouraging direct-to-consumer patients to reach out to their healthcare providers if they are interested in learning more about their genome profile or genetic risk. The problem is that our healthcare system doesn't really do a great job of preparing doctors and healthcare providers with the background that's necessary to unpack that genetic result for patients. Traditional medical school curricula just don't have coverage on these particular topics. And it's led some of the precision medicine advocates to claim that the lack of general knowledge of genetics among medical practitioners is among the most pressing challenges preventing broad implementation of precision medicine. I personally have had some patients who've used 23andMe and got an atypical result present to appointment asking about that result with the paperwork. Fortunately, I have some knowledge of genetics and genomics to assist them, but genetics and genomics and risk prediction are very complicated topics. They usually require about 30 to 60 minutes of counseling time. Most practitioners are given 15 to 30 minutes per patient, so it can really be very difficult to ask providers to conduct counseling on this basis. And this has led to the field of genetic counseling to fill that gap. And genetic counseling is probably one of the most rapidly emerging and lucrative professions in healthcare right now. They're filling that gap that exists between provider knowledge and patient demand. In my field, the idea of pre-implantation genetic testing really gets at precision medicine. And what you see in this video is an embryo that is being held by a suction pipelle for purposes of removing a blastomere. And we'll look at that one more time here. This is an 8-cell embryo. Each of these blastomeres is presumably homogeneous in terms of their genetics, but it gives you the opportunity to take one of those blastomeres, isolate the genetic material, and get genetic readout on that particular embryo. If one of the parents is affected with a particular mutational disease condition, you can screen the embryo for that particular disease condition and determine which embryos are affected and which embryos are not affected. And the unaffected or carrier embryos, which are phenotypically normal, can be transferred back to the patient's womb. So this is referred to as pre-implantation genetic testing for diagnostic purposes. This same technology can be used to screen out embryos that may be affected with aneuploidy. For instance, an extra copy of a certain chromosome, 16, 13, 21, are some of the more common aneuploidies that exist. This is an excellent example of the use of precision medicine for purposes of disease prevention in this instance. But precision medicine can also be utilized for treatment, and that's best exemplified by the field of pharmacogenomics. Pharmacogenomics is a field that uses a patient's genetic information in order to improve the accuracy and or reduce the side effects associated with certain medications. Adverse drug reactions cost the United States healthcare system about 80 billion with a B dollars annually, and that's a lot of money that could be used for other purposes, hopefully to help finance the development of new precision-based medications. When I first developed this talk in 2015, there were 137 medications that had black box warnings from the FDA requiring genetic testing prior to prescription. Now there are 420 medications that have genomic information in their label. We're going to talk about one of them. These are the fields in this bar plot that are actually have the most FDA requiring genomic tests prior to prescription. As you can imagine, oncology leads the way with 184. That subspecialty really is the tip of the spear for precision medicine. And then after oncology, you see other specialties within healthcare that have quite a few medications that have these genomic requirements prior to prescription.
Would you mind clarifying the difference? I'm not sure if they're synonyms, but genomic and genetic?
Yes, good question. So genetic refers to specific changes within individual genes. Genomic is the aggregate of all of those changes. Yep. It's more of a general term than genetics. Okay.
Thank you.
We're going to concentrate on one of the medications in the field of neurology called carbamazepine. It's also known as Tegretol, and it's an anti-seizure medication that was, when I was training, was fairly commonly prescribed. The problem with carbamazepine that we all were made well aware of as young residents in training is that some people are predisposed to a potentially lethal condition called Stevens-Johnson syndrome. Stevens-Johnson syndrome is basically a hypersensitivity or adverse drug reaction that occurs with drugs generally, but seems to be seen in carbamazepine more so than most. And this gives you an idea of the incidence of Stevens-Johnson syndrome with carbamazepine use over the years. The reason that it's not very prevalent after 2012 is because the relationship genetically between carbamazepine and Stevens-Johnson syndrome was discovered around that time. But this photo that came from, clinical photo that comes from Medscape actually shows what Stevens-Johnson can do to a patient. And these are just the cutaneous manifestations. There are also manifestations on the soft mucous membranes internally, and that's where things can get lethal with respect to this particular syndrome. The reason that patients get Stevens-Johnson syndrome is due to their haplotype or the allele at HLA-B 1502 position. If they have the HLA-B 1502 position and carbamazepine binds to that HLA-B position, they develop the hypersensitivity reaction. The reason that carbamazepine binds to both the therapeutic target as well as HLA-B is because of molecular mimicry. On the other hand, other HLA-B haplotypes, versions of that particular gene, don't have that same conformation. So carbamazepine doesn't bind to those HLA-B types, and they don't get a reaction. So the importance here is actually to characterize the HLA-B genotype prior to prescription of carbamazepine. And if the patient has the 1502 type, they should not be prescribed that drug. And this is an excellent example of the power of precision medicine, checking someone's HLA genotype before giving a medication that could potentially lead to a hypersensitivity reaction. On the label for carbamazepine or Tegretol, you can see that there is in the warning label serious dermatologic reactions and HLA-B 1502 allele. So that's an example of a black box warning associated with genotyping. Another example of precision medicine as it relates to taxonomy is really well evidenced by this case. This is a 34-year-old patient who had a 3-centimeter mass in the vulvar area that extended into underlying structures. It was treated surgically twice, and the pathology report from it came back with epithelioid inflammatory myofibroblastic sarcoma, which is a very unusual tumor in gynecology. Had positive margins as well, so that basically indicates that it's a very aggressive tumor. It recurred with rapid growth after 2 months, despite surgery as well as radiation. The tumor genetics on this particular tumor came back with consistent with anaplastic lymphoma kinase rearrangements. And these are not usual in gynecology. They're actually more typical in small cell lung cancers. So the physicians decided that instead of treating the patient with modalities that were typical for vulvar tumors, they would treat it instead as it would be treated as if it were a small cell lung cancer and used crizotinib. And the outcome was remarkable resolution of the mass within 2 months with the use of a chemotherapeutic that typically is used in pulmonary tumors. So what does this show us? It shows us that disease by location or specialty, it's convenient, but it's artificial and limiting. Disease classification by histology even may be limiting in terms of therapeutic response. And moving to a diagnostic classification scheme that is based on the molecular attributes of the tumor may be the right answer moving forward. That is a revolutionary concept in the field of medicine because so many tumors are treated by the site of origin rather than their molecular makeup. So I told you we'd come back to the omics. Let's dissect that out a little bit. When we're talking about DNA, that basic blueprint of 3 billion base pairs, we are talking about genomics. Genomics. When we talk about all of the RNA that is transcribed from those genes in aggregate, we're talking about transcriptomics. And when we talk about all of the proteins that are present in a complex mixture as a result of translation from the RNA, we're talking about proteomics and possibly about metabolomics if we're talking about metabolites from a biochemical pathway. An analog of genomics is a field known as microbiomics. And microbiomics actually studies the DNA. It's also known as metagenomics, a field within metagenomics. And it deals with the DNA of microbes. And a lot of folks don't like talking about microbes, but microbes play a very important role in the human system. And we'll talk about that a little bit in a subsequent slide. I think the omics that is hardest for a lot of the molecular biologists to come by is phenomics. Phenomics derives from the term phenotype. And you may remember from genetics that there's a genotype, in other words, what the genetic code says, and there's a phenotype, which is what your appearance is. And the two are very, very much related. But obviously what you look like or what you appear as may be influenced by environment in as much as it's influenced by genetics. We get a lot of our phenomics from the electronic medical record through a group of folks called clinical informaticists, and they basically correlate molecular signatures with health outcomes. So when I refer to phenomics, I'm really talking about the aggregate of all the clinical information associated with a given patient. So all of these fields are basically, I think I've given somebody the opportunity to draw on the desktop. I told you I was going to touch some wrong buttons, Kate. Basically, all of these omics have given rise to a bunch of new and exciting career fields and have enriched the career fields within other disciplines. And once again, you can see here I've got genetic counselors listed. They are fundamental to the emergence of precision medicine and its application to clinical practice. Okay, so this is my placeholder to remind myself about how much research is done at Montana State. And I am incredibly impressed with how much is done there in the area of STEM and non-STEM research. I understand that last year was, the year 2020 was the year of undergraduate research. When I walked around campus, I saw signs for this everywhere. I know that there was a big conference that was supposed to happen. I think COVID might have changed that a little bit, but there was a big undergraduate research convention that was scheduled to take place there at Montana State. You all have a great location for academic research. And so I thought what I would do in the event that we had some folks on the call that were in the area of research is we talk a little bit about how this field of precision medicine is expected to advance from a research perspective. So let's get into that a little bit. In order to do that, we're going to break this down into different steps. We've been engaged in precision medicine research at my institution here at Madigan for the past 6 years, and we've developed our lab around all of these steps. So let's go through those real quick. The first step in the process of conducting precision medicine research is to detect and define a clinical knowledge gap. Where do we have problems? What clinically would be optimized by a novel biomarker or a molecular therapeutic? So defining the clinical knowledge gap is the first step. The second step is to visualize, based on your clinical knowledge, what the end state solution for the gap might be. In other words, if you don't have access to intra-abdominal fluid, then devising a biomarker within that particular complex mixture is probably not going to translate clinically. On the other hand, if you're able to identify a protein or an RNA signature that is unique to a disease state that can be detected by the bloodstream, that could be a game changer. So visualizing the end state for the intended application. The next step is perhaps the most difficult and time-consuming, and that is to design a biorepository or a biobank with that end state solution in mind. And this is where patience is a virtue, and I mean patience with a C. You have to have them with a T too, but patience is in being able to stay the course over time, because in our experience, we've got a number of active biorepositories at our institution, and they really don't hit biological and statistical power until you've got about 100 to 200 participants in that biorepository. The next is to draft an institutional review board protocol. And this allows us to talk a little bit about the ethics associated with precision medicine and associated research. So we'll get into that a little bit. Then you build the bank with real-time specimen annotation. You consent patients who may want to participate, and when they are going through their clinical care, collect the IRB-approved biospecimens and process those specimens to the molecular level that you're intending to investigate. The next step is to conduct the molecular investigations. That will give us an opportunity in this talk to talk about some of the different platforms that we use in our laboratory to develop precision medicine diagnostics and treatments. The next step is to analyze the data, and that's the field of bioinformatics. And we're very fortunate to have a great bioinformatician at our location. And then, of course, making that discovery generalized knowledge for the benefit of the healthcare at large, and developing it further is the final step. So let's unpack those in a little bit more depth. The first step is to find the gap. Here's a good example, one that I've actually encountered in clinic when I was in training. Mr. Downs has had a rough time with antidepressant side effects. It's too bad we don't have a smarter way to select the best medication for him the first time we prescribe it. And this really gets after the whole concept of trial and error medication administration. This plot from Weiss and Chan shows just how inaccurate healthcare providers are at getting a medication right the first time. There are so many choices. In the field of antidepressants, there's over 20. Which one is the right one for this patient? How do we choose? It's very, very rudimentary in terms of what we're able to do just on the basis of clinical and demographic variables. It's even worse for drugs like hypertension agents, where we're only right 10 to 30% of the time. When we're wrong, there can be serious repercussions. There could be adverse drug reactions. There can be frustration. There can be disengagement from the healthcare system and no treatment at all. There are a variety of different negative repercussions associated with not being right in choosing the medication. So yeah, it would be great to get away from reactive medical care where we make a diagnosis and we continually switch to try and find the right one. Precision medicine promises that we may be able to leverage molecular specificity on the part of Mr. Downs, for instance, to ensure that we choose the right drug the first time. Maybe there's something in Mr. Downs' molecular signatures that allow us to ensure that we choose an agent that not only is going to help with his presenting complaint, which is depression, but also keep him from having adverse reactions to the medication that we choose. And in this slide here, I've given an example of what that translates to. It's the field of pharmacogenomics and how we would set up a study to investigate that further. We'd preferably want to look at the serum or the plasma, and our IRB protocol would be to investigate serum from all patients that we are prescribing antidepressants to, as well as controls, to try and find that signature, that molecular signature that allows us to choose antidepressants more precisely. There are other ways to detect gaps in the literature. They include review articles, consensus conference reports, and one of my favorites is requests for applications for grants, because those are usually predicated on gaps that exist out in biomedicine. So this is an example of a gap in the literature. For instance, if you see this in a review article, despite substantial epidemiologic and clinical evidence for the association. The mechanisms by which cigarette smoke contributes to ectopic or tubal pregnancy are poorly understood. That's a gap. That's your cue that this area, this association needs more study. And so this is a disease pathogenesis study in this instance. And again, I've listed kind of the approach that we would take from an experimental design standpoint to develop that precision diagnostic. Another example is in endometriosis. This is a disease where the latency between presentation with pain and diagnosis in the form of surgery runs about 7 years. That is a condition that is screaming for a biomarker, something to provide a non-invasive that allows us to determine who has endometriosis and who doesn't. So another gap that points to a precision medicine solution. And all of these that I'm sharing with you are active areas within precision medicine biomarker discovery. So visualizing the end state is important. Depending upon what you're trying to do, will determine what layer of the central dogma you really need to probe. And that's going to define what platforms you use in order to investigate it. If it's like a cheek swab, like 23andMe uses, then you're probably working at the DNA level. And that's going to be a certain type of laboratory platform to investigate DNA. Protein is a very different platform, and transcripts are also a very different platform. So you really need to visualize what your end state solution might be that's practical and clinically friendly for patients, and then design your experimental design accordingly. This is an important concept in biorepository development because truth be told, every hospital really is sitting on a biorepository in their pathology labs because they are required clinically to keep specimens that are removed from procedures and surgery in their archives. And these are usually wax, contained in wax called paraffin and embedded in cartridges and maintained in pathology departments. So it would seem that these biorepositories would be excellent stores of tissue available for precision medicine research. But there's a problem with that. And the problem is that with these specimens, there is no quality control in terms of how long the specimen has stayed out of the body. And that is referred to as warm ischemia. And also there's the problem of cold ischemia associated with these because they weren't collected according to a standard operating procedure. The significance of that is pretty important when it comes to molecular signatures, as evidenced by this now fairly dated report from Sprucel et al., where they actually removed a tumor, they took individual biopsies, and they left it out for variable periods of time. They left it out for 5 minutes and then froze. They left another piece out for 8 minutes and froze it, all the way to 30 minutes. Then they took those specimens, processed them for RNA, in this case, the transcript, and then analyzed the integrity of the molecular signature after the specimen had been left out for variable periods of time. And what you see here is that at 5 minutes and 8 minutes, the molecular signature was relatively unchanged. But if that tissue specimen stayed out for 10 minutes or longer, you saw a very progressive decline in the integrity of the molecular signature as a function of ischemic changes within that tissue specimen. That doesn't happen to DNA so much because it's static. But remember, I told you that RNA and protein are dynamic. They react to their environments in terms of the signatures associated with it. And this really highlights the importance of prospectively collected biospecimens for the high fidelity results that you need for precision medicine purposes. So this is one of the biorepositories we keep at Madigan for work in endometriosis. It is very all-inclusive in terms of the types of specimens that are available on a DNA, RNA, and protein level of the central dogma. We can do any level with this particular biorepository. It's one of the largest in the United States right now, and it integrates seamlessly with standard patient care workflows and pathways so that it's not inconveniencing to the patients that consent to participate. In order to maintain privacy and confidentiality of those that participate, every specimen gets a code number instead of someone's name. And this is kind of a hallmark feature of biorepository development. And our collection procedures are in accordance with an international SOP, standard operating procedure, for collecting biospecimens for molecular research and precision medicine in order to assure their integrity. So what about the IRB? Well, the Institutional Review Board is tasked with protecting the welfare of those patients who are generous enough to participate in biorepository protocols. And it's very important that their privacy and confidentiality be maintained at every part in the precision medicine research journey. And so we take great pains, as do others, towards making sure that all of the specimens are annotated without clinical patient identifiers, but rather in an aggregate sense of the word. There's a lot of attention being given to protection of molecular signatures because the DNA code is very, very specific and can be more powerful than even a fingerprint or a retinal scan in identifying who we are with specificity. So many of you may be familiar with the movie Gattaca. If you haven't seen this movie, I highly recommend it. It was a 1997 film. So Kate, have you seen this one? Kate's a film major, so have you seen Gattaca?
You watched it the one night. You watched it the one night I wasn't there.
Oh, okay. Well, I'm going to be interested in your commentary as a film student on this, but it was a 1997 kind of a dystopia, genetic dystopia type film where Ethan Hawke's character is fighting genetic discrimination because he wants to go out into space, and most of the other individuals who are vying for that are a product of eugenics, and they have screening in place to ensure that only eugenically conceived astronauts are allowed into space. And so it really gets after the whole issue of genetic specificity and genetic discrimination. And it's no accident that all of the letters in the title are base pair letters, A, C, T, and G, with the title Gattaca. So I highly recommend that film as a very interesting commentary on the whole idea of genetic discrimination. So a lot of my patients that I work with to enroll in biorepositories are very concerned about this naturally. But fortunately, former President Bush signed into law the Genetic Information Non-Discrimination Act, or GINA, in 2008. And this law prohibits health insurers as well as employers from discriminating potential employees on the basis of an individual's genetic information. So in the very unusual event that genetic information is breached in some way, shape, or form, that cannot be utilized to discriminate against an individual when it comes to employment. As far as specimen annotation is concerned, the clinical annotation is very, very key to ensuring that the phenomic-genomic relationships are high fidelity. And this is an example of one of our biobank specimen annotations. You can see that we track a lot of variables. And our motto is that a biobank specimen is annotated before the sun goes down. So when we are in the OR and we collect a specimen from a patient who's consented to participate, we ensure before we go home so that there's no recall bias or loss of data that we annotate that specimen that very evening. And so we actually fill out a variety of different elements associated with that particular biospecimen, all of which are designed to try and ensure that we get the most signal and the least noise in the relationships drawn from that particular biospecimen. So it's a lot of work, but that work is very important towards making sure that we get clean results from precision medicine studies. So just want to come back to the central dogma cartoon before we talk about platforms. Remember, DNA double-stranded, RNA single-stranded, and protein. We're going to walk through some of the different types of approaches we go through in the laboratory to conduct research at these levels. First, DNA or genomics. These days, it's being done through the use of next-generation sequencing platforms. This is an example of one. This machine can actually read out your entire 3 billion base pair genetic code in 24 hours at a cost of $600. So pretty amazing that you could get that kind of readout in that time at that cost. But that is really what's revolutionized our abilities to make associations between genetics and outcomes.
Now is a dynamic time to be working in the field of microbiology and specifically microbial genomics. One key reason is the rapidly growing understanding of our own microbiota, the community of microscopic organisms that lives on and in us, and the microbiome, the genomes of these organisms. And next-generation sequencing, NGS, is the core technology for metagenomics. Highly complex bacterial communities are commonly identified using 16S ribosomal RNA sequencing. The 16S rRNA gene codes for part of the ribosome found in bacterial cells. Sections of this gene are conserved across the genomes of all bacteria species. But variations within the coding sequence are used to tell different species apart, and even how they're related to each other. 16S ribosomal sequencing is a powerful method for characterizing many different bacteria in a sample. Before next-generation sequencing, this technique was much more limited. With capillary electrophoresis sequencing, each type of bacteria had to be grown as a pure culture, and its genomic DNA sequenced one organism at a time. With next-generation sequencing, many microbial organisms from a complex sample, such as the gut, can be identified simultaneously using the 16S method without the need for colony isolation or culture. Simply swab, perform sample prep, and sequence on MiSeq to get a comprehensive picture of what organisms are in your gut.
This is very powerful technology, and I did want to include that not only for genomics, because that really gets into sequencing, but also microbiomics, which is an emerging field within the field of precision medicine. We actually have more microbes in our body than we have cells. That's a little known fact. And the microbiomic variation between individuals is absolutely huge. Much, much more diverse than the differences between us in terms of our code. We're all about 99% similar in our genetic code, but we're only about 50% similar when it comes to our microbial communities that reside in our bodies. So this is an excellent example of an area where you can leverage differences. And the microbiome is not just an academic community, in terms of demonstrating change. It's appreciated now that the microbiome, especially within the gut, can release hormones and have influences on us in terms of our in terms of mood and other hormonal relationships within ourselves. So a lot of attention is being given to the field of microbiomics, and this is a very powerful strategy to explore that. Transcriptomics gets at the single-stranded RNA, a very dynamic layer of the central dogma. And to probe that, we use machines called light cyclers, which are quantitative PCR machines, as well as the next generation sequencing platform that you saw in the video in the preceding slide. Proteomics is my favorite layer of the central dogma because I really feel that within precision medicine, the platform or layer of the central dogma that is really hitting its stride is proteomics, which describes proteins in our body, the effector molecules. And this is what a large percentage of the laboratory assays that are conducted in clinical medicine are looking for is proteins. This is a machine called an Orbitrap, which is really a tandem mass spectrometry platform that can take a complex mixture and separate it out into its individual components in terms of the proteins that are manifest in that complex mixture. Very, very powerful and a central feature to biomarker discovery. Bioinformatics has taken all that big data and coming up with the clinical insights, the associations that matter between the molecular readout and the phenomics or the clinical data. This is a feature of precision medicine that is a rapidly growing field. And next to genetic counselors, bioinformaticians are the hardest for me to hire because I can't find anyone that there's a paucity of folks out there that can do it. And they are commanding very, very nice salaries as a consequence of this mismatch between supply and demand. So big fan of bioinformatics, and I'm a big fan of genetic counselors. Present, publish, collaborate. I'm just going to share with you. We're getting down to the last 5 minutes of the presentation here, and I thought I'd share with you one of the areas that we are working in at Madigan. This is some surgical photos of a hydrosalpinx in a normal tube. On the left, you can see hydrosalpinx. This is a swollen tube, usually as a result of a infectious disease process such as chlamydia or gonorrhea. Can really wreak havoc on the tubes. You no longer get that fimbriated end that can collect an egg, but rather you get this kind of encapsulated distal end of the tube, and it can result in a 50% reduction in pregnancy rate as a consequence. So we need currently, the only way to diagnose this is through surgery, through images like this, or with equally expensive imaging studies. So we felt that a clinical gap existed in the area of diagnosis non-invasively, and we were looking for a blood test for a specific protein. We went through a lot of different bioinformatic analyses of the fluid within hydrosalpinx and the fluid within normal tubes. And we discovered a protein called mesothelin that looked like it had promise in discriminating between a hydrosalpinx and a normal tube. And sure enough, when we went out into the bloodstream, we found that patients with hydrosalpinx had a very high level of this protein called mesothelin. And those that had normal tubes had much lower amounts of mesothelin in their bloodstream. And we reported on that discovery and are working now towards validating this in a larger group of patients to hopefully develop a test that can be used to discover hydrosalpinx without expensive invasive tests. So that's an example of some precision medicine work that we're doing in our lab. Emerging areas of precision medicine, gene therapy. It's no longer sci-fi to think that genetic therapy will enter molecular medicine. There are estimated to be 10,000 different disorders that are caused by single gene mutations, and those represent targets for genetic therapy. In 2016, the first gene therapy drug, known as nusinersen, was FDA approved for the treatment of a very, very debilitating condition called spinal muscular atrophy. And today there are 16 gene therapy drugs that are FDA approved. Now, this is remarkable and really deals with some of those lethal or debilitating mutational disorders that we've known about for years, but didn't really have much in the way of effective treatment for. Many of you may have heard of gene editing and CRISPR-Cas, which is a prokaryotic technique for editing and the genetic code that is now being used as a molecular scalpel, if you will, to introduce genes at specific locations. Currently, there are 6 FDA-approved trials for CRISPR-Cas mediated treatments. We're going to finish up with some of the challenges associated with precision medicine. Cost. As you can imagine, trying to develop personalized precision medicine-based solutions for patients is very, very costly. The question is, where is the funding for all of these personalized approaches going to come from? To some degree, saving on that $80 billion of adverse drug reaction money could be used to help alleviate cost. But yes, at inception, it will be more costly than beneficial. But scaling also will help to mitigate some of the costs associated with this. Data privacy, we've kind of touched on that, so I'm not going to go into too much more depth on that, but that is a concern associated with the storage of molecular signatures in the electronic medical record or for research archival purposes. Reliability and reproducibility really gets at risk prediction. These platforms that I shared with you are all very good in terms of their reliability from test to test. The interpretation of the changes that are observed is really where there's a lot of variability. And that's going to start to resolve with scale. Finally, translation to the clinical laboratory. As you can imagine, that big tandem mass spec machine is not very clinical lab friendly. So we're going to have to figure out ways to make these tests friendly for laboratory technicians to conduct. And then lastly is generalizability. A lot of the data that we have on precision medicine comes from people of European ancestry. And we really need to change that because when you have data that only pertains to one demographic, it's hard to extrapolate those findings to other demographic populations. And so there is a move adrift within the field of precision medicine to try and be intentional about expanding access to precision medicine studies to all populations.
Vision behind this whole idea is longitudinal. It is collecting information about people over time. Many of our clinical trials don't have enough people in it for us to say with statistical certainty whether the drug has worked just as well for men as it has for women. We live with the laws of averages. And so there are lots of ways in which we as docs can make a huge difference in a more participatory model for clinical research by having 1 million people who have signed up to be part of the clinical research process. One big thing that comes when you have larger studies is you have statistical power to get an answer. With 1 million people, you might suddenly be able to say, might this be something new that we haven't recognized? So I think we will learn things that we haven't dared guess at. We're going to make it lots easier to get answers quicker for important questions. Something new comes along, say a new infection. We have our current one is Zika. Well, you can then pull samples that were put in the freezer 3 years ago and say, was this actually already present? It's going to really change the face of clinical research. My real hope for the precision medicine cohort is it similarly builds a cadre of people who are loyal to the study and proud of participating and eager to continue this donation of information about themselves into the future.
And so the All of Us research program is designed to be inclusive. 1 million Americans will be enrolled in that particular trial. To really scale up and diversify the information that we get to power forward the field of precision medicine. So take-home message, as I hope I've communicated to you, precision medicine has taken the field of medicine by storm and is revolutionizing our approach to patient care and also receiving substantial grant-funded attention from the NIH and other sources. The assays require an understanding of molecular concepts are increasingly permeating our clinical practice. This is scary for a lot of people in medicine currently because they unfortunately just have been a product of curricula that have exposed them to the nuances associated with molecular medicine, but that's changing. Academic medical programs are embracing precision medicine and equipping the next generation with the concepts that will be needed to be versatile in this space. And then well-collected and well-annotated biorepositories are absolutely key towards translating these molecular diagnostic and therapeutic discoveries to personalize and power the future of patient care. So at this point, I'm happy to take any questions. The only one that interrupted me during the talk was Kate. And I could use any questions that you have about things that we covered.
I do have one question. You mentioned there were several drugs that are either verified or in the process of being verified that use the CRISPR-Cas9 system. So do those then perform edits in the body, and if so, on what part of the DNA?
Yeah, that's a really good question. There are 6 that are in FDA-approved clinical trials right now. Most of them are in phase 1. Some are starting to move to phase 2. The disease processes that they are addressing are listed there in that second column. Most of these actually are in different cancers, leukemia, multiple myeloma, among them. And those actually are designed to target the T cell receptor and influence the code within the T cell receptor. Most of these are IV infusion type of approaches. The one that actually is not is for an eye condition, which is a subretinal injection. But the idea behind these is either through CRISPR-Cas or with another vector to integrate the appropriate DNA or nucleic acid to fix an issue associated with that particular disease condition. Does that answer your question?
Yes, thank you.
Okay.
We have a question in the chat. Chloe asked, how should precision medicine respond to disparities between people from an economic standpoint? As in, how will it avoid becoming too elitist?
Yeah, great question. And that really gets at diversity as well. In order for precision medicine to reach the broadest audience possible, it's first important to enroll and to ensure that the results of the precision medicine findings are extrapolatable and generalizable to all populations. And I think that's one of the underpinnings of the All of Us campaign from the NIH. But having the information about precision medicine diagnostics and therapeutics and actually making it available to everybody are 2 different things. The hope is that most third-party payers are going to embrace precision medicine solutions and incorporate that into their menu of covered diagnostics and therapeutics. There's already been momentum in that direction where well-established precision medicine approaches are finding their way onto healthcare management organization lists of covered medical interventions. And so that's probably the best way to make it the most accessible to the broadest population is in that way. Good question.
We have another question in the chat. Chloe says, thank you. Another question in the chat. When looking for clinical knowledge gaps, do you look more to literature or current clinical experience to identify them?
That is a great question. I wouldn't mind having you in my lab this summer. because you definitely have that intellectual curiosity to want to find the gaps, and that's fantastic as a precision medicine researcher. For me personally, I think it's a combination. I have to say that I tend to find most of my gaps through clinical practice. And even though I administrate the research program for our institution and am a principal investigator myself, I never allow myself to stop seeing patients or performing surgery for patients, because my fear is that once I take myself out of that domain, I lose touch with the gaps that are constantly emerging or persisting over time. And so I'm a big fan of actually finding gaps through clinical experience. And I believe that the best translational um, questions originate from the clinical trenches. Now, that said, I have found many gaps either reinforced or de novo through a look at the literature. I shared a couple of those in this talk. Of different gaps that could be identified by reviewing a review article or, the literature associated with a particular disease condition. These are great ways to identify gaps that exist if you don't have access to the clinical trenches as well. A great question. Love
Yeah,
that.
thank you, Cassidy. That was Cassidy in the chat.
Yeah.
I have a question myself. There isn't another question yet in the chat to ask, but I was talking to a girl the other day who was really interested in neurodegenerative
Okay.
diseases. And obviously that's since it's neurodegenerative, the way they look for that for biomarkers at least is more using spinal fluid, right?
Yeah.
And not in the blood because it has to do with neurons. So is the stuff with where precision medicine is right now, is that more focused on blood than it is on neural or spinal fluid, anything else?
Yeah, no, I think that's a great question as far as that end state solution that we talked about. What what do you feel like the gap is going to be best addressed via? And it really depends on kind of what the area is that you're investigating in. If you're looking for a diagnostic test and I spend probably I would say the majority of my time in research in the area of biomarker discovery. So this is an area where we feel that being as non-invasive as possible is best because you're going to have your greatest access and clinical applicability when you're doing blood draws. It's just not feasible really to be diagnostic through spinal taps because they're just not done as commonly. That said, you can get a lot of prognostic information from spinal fluid. And if it's customary for that particular disease condition for spinal fluid to be assayed, then it does present opportunity to look at that mixture for purposes of prognosis or for pharmacogenomics. You may see something in the spinal column or the spinal fluid space that allows you to dial in a better approach to the disease condition. One area that we're doing a lot more in right now is orthopedics, because when the orthopods are working with a joint, they have access to synovial fluid. But what we're very interested in is whether or not synovial fluid analytes can impact or comment on the recovery from injury and help inform, what kind of convalescence may be required for a given injury. Or whether or not that particular joint, if needed to treat with anti-inflammatories, whether or not that synovial fluid comments on the efficacy of anti-inflammatories for that condition. So really, you can use it not just for diagnostics, but also for prognostics as well as therapeutics to dial in on your approach. So it really depends, I guess, on what you plan to do for that neurodegenerative condition and what you feel like the gap is. But spinal fluid may have some benefit in terms of triaging approach.
Thank you. If anyone else has a question. Yes, someone started to go ahead.
Did you hear it, Kate? Because maybe I missed it.
No, I wonder now if that might have just been my echo on your computer.
Oh. Okay.
I got duped. Yeah. Thanks, Dr. Burney, for giving your presentation today.
Well, I sure enjoyed being with y'all, and thanks for having me. I hope that get a chance to participate in some of the other honors presents, and wishing y'all a great evening.
You're getting lots of thank yous in the chat.
Oh, well, thank you. My pleasure. Kate, just make sure I get my 700.
Champ change.
Yeah.
I will make sure of it.